Hello, welcome back to Systematic Symphony of Species. In this video we are going to talk about conceptualization of species with a genomic approach. The paper, A Genomic Conceptualization of Species, by David A. Liberals discusses species concepts from a genomic perspective. The author highlights the empirical nature of existing species concepts such as biological, phylogenetic, sequence distance, observable phenotype, and ecological species concepts. Liberals introduces a theoretical perspective on species based on the genotype-phenotype map, emphasizing the relationship between genomic data and species classification. The author explores the vast space of possible genomes and their connection to observable phenotypes, discussing the necessity of a mathematical framework to define species boundaries and maintain monophyly as a core principle. The paper also addresses the applicability of this genomic conceptualization to bacterial lineages and the potential for its realization through advances in machine learning and genomic studies. The author concludes by emphasizing that each species is defined by a large set of possible sequences and the importance of understanding the connectivity of viable genomes for speciation and sequence diversity. The paper provides a comprehensive overview of species concepts from a genomic standpoint, offering a unique perspective that encompasses various existing species definitions. The author effectively presents the challenges associated with traditional species concepts and proposes a theoretical framework based on the genotype-phenotype map, which takes into account the vast space of possible genomes and their connection to observable phenotypes. Liberals underscores the need for a mathematical approach to define species boundaries, particularly in maintaining monophyly, and discusses the potential application of this genomic conceptualization to bacterial lineages. Moreover, the commentary anticipates the future realization of this approach through advances in machine learning and genomic studies, highlighting its potential to redefine species classification in various contexts. How does the author propose using a theoretical genotype-phenotype map to conceptualize species? The author proposes using a theoretical genotype-phenotype map to conceptualize species by linking genomic data to species classification. This approach is based on the understanding that for a genome of size 10 to the power of 9 base pairs, there are approximately 4 to the power of 10 to the power of 9 possible genomes, describing all genomes of precisely 10 to the power of 9 base pairs. The author introduces a theoretical perspective on species based on the genotype-phenotype map, which aims to encompass various existing species concepts. The theoretical genotype-phenotype map is depicted as a geometric representation of species in a high-dimensional space, where each species is represented by a different hypersphere-like volume containing all possible genomic sequences that give rise to that species. The author emphasizes the importance of maintaining monophyly as a core principle in defining species boundaries using this approach. Additionally, the author discusses the potential applicability of this genomic conceptualization to bacterial lineages and highlights the possibility of defining mathematical properties about species described in genotypic space. The author also suggests that advances in machine learning and genomic studies could lead to the actualization of this approach in defining species in various contexts. What are the limitations of empirical species concepts, and how does the proposed theoretical perspective address them? The limitations of empirical species concepts lie in their reliance on observable characteristics and clustering of individuals, which may not fully capture the complexity of species boundaries. These concepts, such as the biological, phylogenetic, sequence distance, observable phenotype, and ecological species concepts, are based on empirical observations and characterizations of individuals. However, they may not be robust in defining species boundaries, especially in cases of phenotypic plasticity and in the context of bacterial lineages where traditional species concepts may not apply. The proposed theoretical perspective addresses these limitations by introducing a genomic conceptualization of species based on the genotype-phenotype map. This theoretical framework aims to provide a more comprehensive and mathematically precise approach to defining species boundaries. By considering the vast space of possible genomes and their connection to observable phenotypes, this approach offers a more nuanced understanding of species classification. It emphasizes the importance of maintaining monophyly as a core principle in defining species boundaries and introduces a geometric representation of species in a high-dimensional space. This theoretical perspective also has the potential to define mathematical properties about species described in genotypic space, providing a more comprehensive and precise framework for species classification. Additionally, it addresses the limitations of empirical species concepts by offering a theoretical approach that can be applied to bacterial lineages and other contexts where traditional species concepts may not be fully applicable. How does the concept of species boundary relate to the genotype-phenotype map and the idea of monophyly? The concept of species boundary relates to the genotype-phenotype map and the idea of monophyly in the context of defining species based on genomic data. The genotype-phenotype map provides a theoretical framework for understanding the relationship between an organism's genetic makeup and its observable characteristics. In this context, the concept of species boundary is linked to the genotype-phenotype map by considering the vast space of possible genomes and their connection to observable phenotypes. The theoretical perspective on species, based on the genotype-phenotype map, aims to provide a more comprehensive and mathematically precise approach to defining species boundaries. Furthermore, the idea of monophyly, which refers to a group of organisms that includes an ancestral species and all of its descendants, is crucial in defining species boundaries using the genotype-phenotype map. The author emphasizes the importance of maintaining monophyly as a core principle in defining species boundaries. This means that when defining species based on genomic data, it is essential to consider the evolutionary relationships and genetic relatedness among individuals within a species. The theoretical perspective on species, rooted in the genotype-phenotype map, takes into account the connectivity of viable genomes, the expected speciation rate, and the sequence diversity available to sample within a species. This approach aims to provide a more nuanced and precise framework for species classification, considering both genetic and phenotypic aspects. What are the potential implications of this theoretical approach for defining species and understanding genetic processes? The theoretical approach proposed by the author, based on the genotype-phenotype map, has significant implications for defining species and understanding genetic processes. By considering the vast space of possible genomes and their connection to observable phenotypes, this approach offers a more nuanced and mathematically precise framework for species classification. It addresses the limitations of empirical species concepts by providing a theoretical perspective that can be applied to bacterial lineages and other contexts where traditional species concepts may not be fully applicable. One potential implication of this theoretical approach is the ability to define species boundaries in the absence of pure observation. By linking genomic data to species classification, this approach allows for the potential definition of mathematical properties about species described in genotypic space. It also provides a framework for understanding the genetic processes underlying phenotypic diversity and species classification. Furthermore, the theoretical approach has the potential to contribute to the understanding of evolutionary processes and speciation. It can describe both the expected speciation rate out of a particular species over evolutionary time as well as the sequence diversity available to sample within a species. This can lead to insights into the connectivity of viable genomes, the evolutionary pathways leading to speciation, and the genetic mechanisms driving phenotypic diversity. Overall, the theoretical approach based on the genotype-phenotype map offers a comprehensive and precise framework for defining species boundaries and understanding genetic processes, with potential implications for evolutionary biology, population genetics, and species classification in various contexts. In conclusion, Liberal's paper offers a thought-provoking insight into species conceptualization by introducing a genomic perspective. The proposed theoretical framework based on the genotype-phenotype map presents a novel approach to defining species, addressing the limitations of traditional species concepts and emphasizing the potential for mathematical properties about species described in genotypic space. The commentary encourages further consideration of this conceptualization in defining species and underscores its potential implications for understanding the genetic processes underlying phenotypic diversity and species classification. Thank you for watching. Please share your comments on the genomic approach for species conceptualization.